evolution of protein synthesis and that of intelligence. The intelligence of life had evolved significantly due to wide variety of proteins. This presentation will describe that the outline of replication of protein had been elucidated by the understanding that chirality is involved, and it advanced the understanding of digital aspects of intelligence. Amino acids of the same chirality are peptide bonded to form the first protein. The protein is made up of amino acids. The amino acid is composed of a carboxyl group COOH, an amino group NH2, a side chain R and hydrogen H as shown in Fig. 1. When amino acids of the same chirality are peptide bonded, numerous types of proteins with a helical structure are produced. Professor Kenji Ikehara has proposed the GADV hypothesis as a protein world hypothesis. The protein molecules those are embodied in living organisms by natural selection include structural proteins in which side chains of amino acids attach to cell membranes composed of long-chain hydrocarbon molecules to strengthen biological tissues, and enzyme proteins that promote reactions. However, those protein is composed of a huge number of combinations, and it is not easy to elucidate the function from atomic arrangements. The current proteins are produced by the RNA that is generated from DNA, and tRNA carries an amino acid and a protein is synthesized intermittently in amino acid units with reference to the genetic information of mRNA. Therefore, this presentation tried to elucidate the origin of the mechanism of protein production based on the knowledge on current studies. The double helix structure of DNA shows how to accurately match different chiral amino acid pairs. The living organisms is maintained by multiple elements. Those perform synchronized reactions in harmony with the surroundings. There are two types of elements in a control system. The controller and to be controlled elements must be distinguished. The amino acids those make up living organisms are left-handed type L form, and the sugars those make up the nucleic acids which carry genetic information are the right-handed type D form. Fig. 2 shows a model of Watson-Crick DNA. DNA is composed of molecules with a helical structure that is paired with right-hand helix and left-hand helix. In double helix structure, where the direction of shift on a right-handed helical structure and left-handed helical structures of amino acids is opposite, the gears of the amino acid units are precisely segmented. However, atomic arrangement of amino acid of L form and that of D form cannot match due to different chirality. Therefore, a codon with 3 bases is attached to the amino acid of L form, an anticodon is attached to the amino acid of the L form, and the amino acid is identified by the complementary relationship between the codon and the anticodon for individual amino acid. Forming mechanism of DNA that is indicated by the structure of DNA. As shown in Fig. 3, the double helix of DNA has the leading strand and the lagging strand of different chirality. Both chain-like molecular sequences synthesize in the 5' to 3' directions, as shown in Fig. 3. Therefore, the molecular sequence of the helical leading chain and lagging chains proceeds in the reverse winding direction. Since a unit of the lagging chain contains D-form chain, each amino acid is attached by short L form of Okazaki fragments. When the two main chains of DNA are separated into an L-shaped leading strand and an inverted D-forming lagging strand, the separated D-shaped lagging strand is broken into amino acid units, with which a short L-shaped Okazaki fragment attached to the front and back, and moreover L-form amino acids attached to form tRNA. The overall structure of tRNA will be matched with L-form of the leading strand of mRNA. This interaction could be assisted by the antagonistic and inversely progressive effects present in the organized structure in a helical structure system, as noted by the author. Structure of DNA and RNA. Amino acid molecules attach to cell membranes whose main components are long chain of hydrocarbon molecules, and the amino acids form peptide bond to form protein molecules, and some of cell membranes to which the protein molecules are bound becomes an enzyme. On the other hand, sugar chains adhere to the biological membranes that make up the surface of the cell. The sugar is involved in the interaction between cell membranes and protein molecules. Nucleic acids are molecules that carry the genetic information of living organisms, but RNA is the sugar part of the nucleic acid is ribose, and DNA is 2-deoxyribose in which the hydroxyl group at 2' position of ribose is replaced with a hydrogen group. The nucleic acid is the information media for protein replication. At replication of a protein, tRNA carrying amino acid molecules is used to intermittently synthesize amino acid units with reference to the genetic information of mRNA. Structure of the ribosome that replicates protein. Fig.6 shows a model of protein replication that takes place in ribosomes. It indicates primitive protein replication in a cell membrane. Duplicate fork is moving from right to left. The matching of amino acid is carried out intermittently as amino acid unit. Since protein molecules need to reproduce strictly the sequence of amino acid units, one amino acid molecule is matched frame by frame as if it corresponds one teeth of a gear, and the step is shifted to perform the next process. Thus, the replication of protein molecules had been accomplished through step-by-step processes. Principle and evolution of intelligence. Here, if the unit of activity to be controlled is the reaction of an element of activity that becomes an impulse, then the change in state due to that activity is digital. In mathematics, the relationship between impulse and digital as a function of time is the relationship between differentiation and integral, as shown in Fig.7. It is not possible to describe the intermediate course of the reaction of the unit, but the change in the state of the unit can be described as a transition of the state before and after the reaction. It is thought that state transitions are controlled by memorizing the state changes before and after as a causal relationship and transferring the rules. Incidentally, Heraclitus from 540 BC to 480 BC argued, everything is flux, i.e., the laws of life and the universe are change, and man cannot escape from them. Every cell's inner life must respond in cooperation with the surroundings at each state transition. The ignited group of reacting elements disappears at the next moment, and the next group of reacting elements ignites in response to the newly ignited group of reacting elements. Since the reaction element of the next moment ignites according to the pattern of the ignited reactions, the perception of reality is made by the pattern recognition. The reactive elements of living organisms have evolved in survival in harmony with the surrounding conditions. In the network of biological reactions, the reactions that are not in harmony with the surrounding situation are eliminated. The development of intelligence due to the addition of circuits. The circuit that transmits the reaction becomes robust by repeating the activities. By accumulating experience, the network for transmitting information is expanded, and the information processing function was improved by the additional circuits. Between neurons in the cranial neural network, axonal terminals of presynaptic cells transmit information by contacting the dendrites of postsynaptic cells. The connection of the synapse results the formation of intelligence. When young children learn to use words, their understanding of things progresses. Human beings have come to use language as an information medium, and the memory of things has become certain. With the addition of a network of linguistic expressions, the coordination between the elements of activities makes higher level of intelligence. Now, the area of the associative cortex that uses human language has developed and occupies 2/3 of the entire neocortex. Human being thinks about the meaning of state change. In movies and other moving images, images that are provided intermittently frame by frame are recognized as continuous images. The neural networks control behavior through the transfer of intermittent impulses. The steps of each operation can be described using state transitions before and after. In other words, it can be described by the logic of if-then rule, that, if A equals B, then X equals Y. Human beings perceive digital state changes as analog activity and understand the reasons for digital transitions as causal laws. Here, the meaning of the activity can be understood by how the whole activity that the activity has passed has affected the reality. Human beings understand changes in state by the law of cause and effect, but it is a closed world in which the logical world is applicable only to humans. Therefore, it is necessary to value the facts of the real world without prioritizing the world of thought. Summary: the control mechanism must distinguish between to control and to be controlled. Amino acids are composed of left-handed L form for chirality, on the other hand, right-handed D form for sugars in nucleic acid. Synthesis of protein proceeds intermittently in units of amino acid molecules. The mechanism of responding digitally has made it possible to processing information accurately, so it has been possible to develop into a high level of intelligence. Here, thinking has a role of controller. It is natural for the world of thought to differ from reality. It is necessary to avoid falling into the logic of only humans in the competition between humans. In such case, it is helpful to recall that the intelligence of living organisms was born under the conditions to harmonize with the surrounding circumstances. Thank you for your attention.